A genetic tweak that improves the accuracy of protein synthesis can lengthen an organism's lifespan, according to a paper published this week, September 14, in Cell Metabolism. The results were consistent across 3 species, the nematode Caenorhabditis elegans, the fruit fly Drosophila melanogaster, and the yeast Schizosaccharomyces pomba, suggesting that building better proteins may be linked to longevity in other species, too. The work is very convincing, very compelling, and I think it addresses a really important outstanding question in the field of aging. What are the best ways to look after our proteins and help ourselves to function better for longer? Says protein researcher John Labadia of University College London. For example, protein production, folding, and degradation all decline in quality, such that loss of protein homeostasis, proteostasis, is a major hallmark of aging and age-related diseases, says molecular biologist Patrizia van Osten Hall of the University of Leeds, who did not participate in T. In the case of protein production, the translation of RNA code into peptide chains, the prevalence of errors, correlates positively with lifespan. University of Rochester aging researcher Vera Gorbanova, who was not a part of the research team, writes in an email to the scientist. However, evidence showing that one can take a short-lived organism and extend its lifespan by making translation more accurate was lacking, she writes, adding that the new Cell Metabolism paper provides this last missing proof. Within the protein factories of the cell, known as ribosomes, a PR. RPS23 was thus the obvious candidate when looking for ways to improve fidelity, explains molecular biologist Ivana Biedov of University College London's Cancer Institute. Within the protein, they found a region so highly conserved that, with the exception of some microorganisms that live in extremely hot environments, all species had an identical amino acid at the same position. What are the best ways to look after our proteins and help ourselves to function better for longer? John Labadia, University College London. Biedov wondered how the single amino acid change seen in thermophiles might affect translational accuracy. Her team introduced the change into the RPS23 gene of D. melanogaster and found that not only was translation accuracy improved, the flies also could survive higher temperatures and lived roughly 10 to 20% longer than control flies. Biedov's collaborator, Felipe Cabrero of the MRC London Institute of Medical Science, then introduced the same RPS23 amino acid change into C. elegans and got similar results. Further studies showed the amino acid change boosted translation accuracy and lifespan in yeast as well. Finding consistent results across 3 species makes you more reassured about what you're seeing, says Cabrero. It's a robust observation. In the flies and worms, the life-extending mutation was associated with delayed development and reproduction, though ultimately, the numbers of progeny produced were similar to those of control animals. Similarly, in yeast, the mutation caused colonies to grow more slowly. It is not clear why the mutation caused this developmental delay. Translation rates between test and control animals appeared to be equivalent, as did other phenotypic traits. But the delay could explain why the mutation, which is otherwise seemingly beneficial, isn't more widespread in nature, say the researchers. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.